Good.
Okay, ready? Hi everyone, how you guys doing? Okay, so today I'm happy to introduce Dr. Ben Domingue, who joins us from Stanford. He attended the University of Texas at Austin for both his bachelor's and master's degrees in math. After that, he went on to the University of Colorado at Boulder, where he received his PhD in research and evaluation methodology. And during this time, he was the recipient of a quantitative dissertation award from the AERA. Ben is currently an assistant professor at Stanford's Graduate School of Education, as well as a research associate with the Institute of Behavioral Science at the University of Colorado Boulder. His research interests include how student outcomes can inform our understanding of student learning, teacher performance, and program efficacy. Additionally, he's interested in the relationship between improved measurement of human DNA and our understanding of human behavior. Please join me in welcoming him. Thank you so much.
So thank you all for inviting me, and sorry about the delays. I will say at Stanford we never have IT problems. It's actually very not true, but I can see it. Okay, so the talk today is titled Genetics and Education, Recent Developments in the Context of an Ugly History and an Uncertain Future. I obviously don't know most of you, so we'll see how this goes. I hope this talk brings you up to speed on what I think, and hopefully can convince you of, is a kind of pressing area of science. Whether you wish it was happening or not, I think it does behoove us all to know a little more about it. I'd like to start by motivating the talk based on this paper, or this article that came out 2 years ago in the Atlantic. The title is The Genetics of Staying in School. This is a kind of big, high-profile thing written up in the Atlantic. Here are a bunch of other examples. This is, the NIH saying, look, we've started to identify genetic links to educational attainment. This is something that Carl Zimmer wrote for the New York Times in January that's on a similar theme. This is from the Smithsonian based on some work on Iceland and education genes. This is a small sample. You can find lots of this kind of stuff in very mainstream media outlets. Where's this all coming from, right? So the genesis of pretty much all four of those newspaper articles is this paper that was published 2 years ago, May 2016, in Nature, right? So this is kind of science at the absolute highest level. And the title is Genome-wide Association Study Identifies 74 Loci Associated with Educational Attainment. Now, I should note right up front, I'm not involved with this paper. Right? This isn't, you may or may not like the work these people are doing. That's fine. I have nothing to do with that. I am doing work kind of downstream of that. I saw stuff like this start to come out, and I got very interested in this. What do we actually make of this? I was originally trained as a psychometrician. In fact, I guess I'm kind of the grandson of Mark Wilson. My advisor went to school here, and so I think about my work as kind of a sequence of validity studies, asking kind of questions about how seriously we take the results reported in studies like these. And so, at this point, to my way of thinking about the world, Pandora's box is kind of open. This is a tweet from the same group that published the article on the previous page, just basically announcing the forthcoming publication of a new study along these same lines. That should be out kind of any day now. I'm going to show you at the end more kind of studies in this vein that look at not educational attainment, but other disorders or characteristics that might be of relevance to education. So I think that this is just going to be the world we live in now. And so here are the things that I'd like to try to do in the talk. I want to first give you a bit of an overview of molecular genetics and the kinds of things that people are doing now and how they differ from the past. I want to talk about the GWAS concept, right? This is kind of pivotal, and it's the way we link genotype and then observe traits or behaviors. And finally, I want to give you a kind of survey of findings informed by educational attainment GWAS. So this Nature article a few slides ago, I want to tell you about what we've learned through some follow-up studies. The hope is that you'll leave here ready to engage with this literature as you see fit. Being in a school of education, I understand that people have very critical stances on some of this stuff. That's fine. My goal is to hopefully give you a better kind of point of entry into reading this literature and understanding. I will talk about some of my work, but I'm going to largely focus on the field and kind of gesture in the direction of my work in particular at certain points, but I'm going to try to cover things above and beyond just what I do. I also want to take a moment to kind of situate the talk, so especially at Berkeley, some of this maybe has kind of ominous echoes. And so I want to tell you how I think it's a little bit different. So I think genetics is a really powerful lens for studying human development. And the reason why is that unlike nearly everything else we deal with, we have crystal clear information on what causes DNA. It comes from your parents. You get it at conception. It's not changed by anything that happens thereafter, right? That's a really powerful tool. Everything else in social science is so much messier than that. It is the case that DNA will be associated with the kinds of things that we also care about, and that'll muddy the waters. But I do think in terms of studying human development, the fact that it's unchanging across the life course can be a really powerful mechanism for understanding why humans end up how they do. And in particular, I think genetics are a good way of potentially studying how individuals who begin in relatively similar situations end up different. The classic example here is siblings, right? So you can find a century's worth of twin literature where they look at differences between MZ and DZ twins. Lately, we've been doing more things where we just take siblings who, they start from very similar environments in terms of the kind of the social world, but they differ genetically, right? And so the difference in genes there, it can be a powerful tool for understanding how siblings end up different. Genetics are never the tool that I would think to use to explain why people from kind of manifestly different starting points don't converge, right? I think that's kind of crazy. This is not what I'm going to be trying to do. And the last thing I want to say about this, nothing that I'm going to say challenges the idea that powerful social forces have very kind of potent consequences for individuals. I certainly believe that to be the case. The things that I do are orthogonal in a lot of cases, or perhaps interact with these things, but this is definitely true. Genes don't negate this point at all. Furthermore, just because a trait is genetically influenced does not mean it's immutable or that we can't do anything about it. I like to think about this through an example of BMI. So body mass index is something that we have a pretty good understanding of its genetic architecture. People are kind of developing drugs based on genetic studies. How your body is shaped is strongly genetic. And yet, in the last 30 years, we've seen a complete sea change in obesity rates in this country. That's not genetic, right? That's all social stuff. That's all environmental. So just because things are genetic, just because I'm going to say this is heritable, this is genetically influenced, that's not to say that it's immutable, that policy can't do anything about it. So this is kind of the throat clearing. I'm happy to, I don't know what the norm here is. I'm happy to take questions as they come, if that helps. Are there any questions at this stage? Okay, well, if they come up, please just let me know. Okay, so the starting point for all of the things I'm going to talk about are the completion of the Human Genome Project. And one thing I want to emphasize is that the Human Genome Project terminated and kind of culminated in 2003, right? So we're really just 15 years into the kind of modern age in terms of what it means to be able to study genetics. When I go to the doctor and hear them talk about genetics, it's very clear that they were trained before 2003. Right? This is very new stuff that we really did not understand. If you go back to 2000 and ask a geneticist how the world was going to work, they would have gotten everything wrong, right? We've discovered very interesting things that we just didn't kind of foresee, and so the novelty here and the newness, I think, is really important. One of the kind of key factors behind everything I'm going to say is the fact that the genotyping cost has streamlined, just plummeted, right? They're now down here in the kind of well, I don't know, you can send your spit to 23andMe for $100, right? Way faster than Moore's Law these declines have been. So we now just have so much data. And I'm not saying that's a good thing or a bad thing, it's just a fact. And with that data, people are going to want to make kind of claims, right? So we need to think carefully about it, but we can't get away from the fact that there is all this genetic data running around. And you can see papers being published by 23andMe, by Ancestry.com. This is a very kind of high interest area of work. Okay, so let me tell you a little bit about DNA. We're only going to look at a few slides, but I think it's going to capture the kind of key stuff that you really need to know. So presumably everybody has seen the kind of the double helix, right? Where you have all these kind of what I like to think of as ladder legs running across. These are base pairs. Across the entire human genome, there are about 3 billion base pairs in total. And humans are identical at the vast majority of them. At the vast majority of them, in fact, there's no variation, right? Like, we kind of all have the same genotype at a location that came from the kind of, farthest back ancestor that you'd like. But every now and then we see a mutation occur, right? And so here are two kind of pieces of DNA. This is the same piece. You can see that we have AT, etc. And then here we have a point of variation. This guy is an AT, this guy is a GC. This is known as a SNP, or a single nucleotide polymorphism. It's a single change in the kind of alphabetic code that is DNA, and it's a change in one location. Other more complicated forms of genetic variation exist, but most of the action, especially in terms of translating stuff downstream in the social and behavioral science, is working with these SNPs. SNPs occur roughly 1 in every 300 base pairs, right? So at the vast majority of these points, there's no variation. About 1 in every 300 base pairs, you get a spot wherein there's been a mutation at some point in the distant past, and we typically only study variation that is relatively common. We don't study something that, a kind of de novo mutation that just you possess, right? This is something that occurred generations back and is now kind of filtered out such that one large segment of the population has one flavor of DNA, and the other segment has the other section has the other, okay? Now, SNPs are not genes. So genes are in fact protein coding regions of DNA. So a SNP is like a house, a gene is like a neighborhood or a city, right? A gene is like a large region of DNA. We have, as I mentioned, about 3 billion base pairs, only 25,000 or so genes. You can find, there are types of rice, for example, that have orders of magnitude more genes than we do, right? So that is one of those facts that if you went back to 2000, geneticists didn't know how many genes we had and would have not guessed the number in the right ballpark, right? They would know orders of magnitude, just had no clue. So we don't have that many genes. SNPs may be embedded in genes, right? Maybe this is a SNP. Maybe we have SNPs in these kind of regions that aren't associated with any gene. That stuff's all messy, and for reasons that I'll get to later, I don't think it's incredibly important for a lot of my work, certainly, and in fact for our broader understanding of genetics, at least in this kind of colloquial way. Okay, so the two flavors of DNA are called alleles, right? So you have a major allele and a minor allele. The major allele will be the one that's more common in a population. The minor allele is the one that's less common. You'll also hear people talk about the reference allele. So maybe I'm interested in how many copies of the GC allele you have. Right? I would count those. We get this stuff from mom and dad, like I said. That's very important. And what we typically focus on is the number of reference alleles you have at each of these SNPs, right? So at each of these points where there is variation, we just are kind of arbitrarily counting how many of one of the variants you have. So maybe we're interested in AT, so you could have 0, 1, or 2 copies of the AT allele. Neither your mom or your dad gave you one of your parents gave you one, or both of your parents gave you AT, right? So you could have a 0, 1, or 2. And this yields this kind of data, right? So I have here a kind of cartoon of what this data looks like. We have about 1,000 people. In most of the kind of panels that we do now, we have about 1 million SNPs. Every element of this matrix is just counting the number of reference alleles at each SNP. So this guy has none of the reference alleles. This guy has 1, this guy down here is 2, etc. Right? This is not like wildly out of the realm of understanding for people that deal with data. When I started this, I was a psychometrician. I thought, this looks like I had response data, right? I have this guy got no points, this guy got 1 point, this guy got 2 points. And I had some intuitions about the types of things you could do with that data. So for example, you might think about data reduction in terms of principal components analysis. And that turns out to be a very powerful idea that I'll show you a little bit more about momentarily. The key question is, how do we tie this large set of genetic variants to traits? Right? I want to figure out which of these SNPs is driving variation in some kind of disease, behavior, anthropometric trait, right? I want to try to tie my genotype to my phenotype. And the way we do that is known as a genome-wide association study. This is kind of the approach that has been adopted by the biomedical establishment, and it's what they do to understand the link between genotype and phenotype across pretty much every disease you can imagine, every type of human behavior. This is the tool. And it's really a very simple tool. So the first thing you do is you assemble lots of data. You get as many data sets as you can that have both genes, right, that have people genotype, and that have information about the phenotype of interest, right? So maybe that's height, that's pretty easy. Maybe it is smoking status, whether you smoke, how much you smoke. It becomes trickier if you want to study something like Alzheimer's disease. You know, if you have a genotype sample of younger people, none of them are going to have Alzheimer's, presumably. People don't typically live too long after they get Alzheimer's, they die, that becomes a problem. But this is the name of the game. We get as much data as we can that consists of genotype and phenotype. I then just regress the outcome on each of the individual SNPs plus some controls, right? So I would take this vector, right, this one column right here, and I would regress my trait, right? This first guy, maybe he's 5'9", right? That'd be the outcome, this would be the predictor. Second guy is 6'0". This is our predictor, right? And I regress my column of outcomes on this column of SNPs. And then I just kind of do this ad nauseum. I do this for every SNP, right? I do this across my million markers, or however many I have. And then I kind of, worry about the fact that I've done multiple testing of the worst kind here, right? So I have to do some corrections for multiple testing. And so people tend to talk about loci that have, or SNPs that have p-values under 1 × 10 to the negative 8. So they look for those SNPs that are really highly associated with the trait.
Do you use machine learning for this?
People are kind of starting to do that kind of thing.
Okay.
But they started with this kind of like dead stupid thing that has worked fairly well.
Okay.
And so this, as I said, has worked fairly well. So here are 2 pictures. On the bottom we have continuous outcomes. On the top we have continuous outcomes. Down here we have diseases. In both cases, as they assembled larger samples of people, they got more and more hits, as they're called, more and more loci that they could tie genotype to phenotype, right? So breast cancer is down here, so you can be sure that the BRCA gene is showing up as a hit in this kind of GWAS. I don't know if they have Alzheimer's here, they don't, but if you heard about Alzheimer's, you may have heard of APOE. This is a very common, or a very well-studied genetic mutation that's highly predictive of Alzheimer's. Those are the kinds of things that we've identified via GWAS. You oftentimes see the results of GWAS given in terms of these Manhattan plots. Manhattan kind of reflecting the fact that you have these buildings, so to speak. What we have here on the x-axis is basically genetic distance, kind of genetic location. The different colors are different chromosomes. Here's chromosome 7. And so basically what you have is that the buildings represent a point in the genome that seems strongly associated with the outcome. Right? So right here, this blue tower, we're definitely getting something here on chromosome 3 that's really kind of associated with variation in outcome. So that's nice. And in fact, we've kind of played this game long enough that we have discoveries throughout the genome linking all kinds of complex diseases and pretty much anything you like to different regions of the genome, right? So any kind of relatively common disease would probably have like a well-done GWAS on it by now, any anthropometric trait. And we're starting to use these findings to drive both diagnosis and therapy development. So you can find people, for example, who are very interested in the idea of using GWAS results to learn, or basically to better predict who's going to get heart disease. Today I was reading about people that want to do this for type 2 diabetes. Right? So that's one thing that you can do. Other people are interested in developing drugs. What I do is different. I'm interested in how we interpret these findings in the messy wild, right? Humans are not lab rats, they're very misbehaved. And I think we're going to have to account for a lot of that to really understand what's going on here. And so I'm going to talk more about that in the slides that come. Now, before I get there, I want to tell you about a few kind of troubling, well not troubling but kind of things that have proven difficult. So one major limitation is that genetics and human history are very closely kind of linked. They're intertwined in the most complicated of ways. One illustration of this is this picture from an article by John Domingue. This was a large part of the reason that he won the MacArthur Award this last go around. And what we have here is a map. We have an actual map, and then we have a map from DNA. So what he did, he took the kind of SNP data I showed you earlier. He then computed principal components from it. So if you've computed principal components, that's presumably something you can start to imagine doing. He plotted PC1 here, the north-south axis, PC2 here, and he plotted people and he color-coded them by their country of origin and he gets so much information about the map of Europe from his DNA. Right? This is really rather incredible. Right? So you have the Spaniards and the Portuguese over here. You have France, Ireland, Great Britain, Scotland, Belgium, Germany, and the rest of Europe. You have Italy down here. I've always wondered if these people were from like Sardinia or Corsica or something. But human history is kind of deeply woven into our genes. Right? These are relatively homogeneous Europeans. I think in this sample, to be in this sample, not only were you kind of Italian, but your grandparents were Italian, so you weren't. They did not include more recent, say, immigrants from Ethiopia or something like that. And one punchline of all this work is that genetic discoveries from a sample of one ancestry do not easily translate to genetic discoveries from a sample of different ancestry. It's not that we think the biology is fundamentally different. There is a lot of messiness that goes on there. So most of the things that I'm going to talk about today are going to be based on studies of people of European ancestry. That's a real limitation that I'll address at the end. Another key finding from the first decade of GWAS studies is what's known as polygenicity. So here's one statement of this idea. It's been called the fourth law of behavior genetics. A typical human trait, I think you can just kind of scratch out behavioral, I think this is just generally true. A typical human trait is associated with a great many genetic variants, each of which accounts for a very small percentage of the behavioral heritability. So if you took high school biology, you probably remember Mendel's P's, and something was either on or off, right? If you got one allele, you had the kind of smooth shell. If you got the other allele, you had the wrinkly shell. It turns out that almost nothing is like that. There are a few examples. So cystic fibrosis is like that. If you kind of get the deleterious mutation, you end up with the disease. If you do not, you don't have the disease. But almost everything else is much messier, and it's being driven by variation all across the genome. I had a person at Stanford tell it to me this way. He said, when I go to the hospital, the things that I see that are putting people into beds are due to polygenic traits, not simple traits, right, that have simple genetic architectures. So this is part of the reason that I think, I think in some ways this actually calls us to do more work with genetics, because the geneticists are going to need our help. They are stuck with very weak signal, and I think they're going to need our help parsing it. And I'll show you some examples as to why later. And the biologists are starting to realize that this is a real problem too. This is a paper from last summer by Jonathan Pritchard and colleagues, where he talked about traits being not just polygenic, but omnigenic, right? That the genetic influence is so pervasive from across the entire genome that they're having to kind of fundamentally rethink how they understand DNA. And so this is all it was a very shocking development to the biologists, at least some of them. And the way I kind of deal with this problem is through the use of what are known as polygenic scores. So basically what I do is I take my genome-wide data, I showed you this picture earlier. I take these results are results from a GWAS in the form of a Manhattan plot, and I think about these as weights. So I take this SNP number 2, I find it in my Manhattan plot, maybe it's this little point right here. And I'm now just going to basically add up the number of alleles you have where I use this as my weight. And so if I am talking about, right over here where I have kind of genes, genetic variants that are very predictive of the trait, I'm going to really count it when you have those risk alleles. If I have something very low level, I'm going to downweight that. And I'm going to do this to construct a nice, continuously distributed predictor of whatever outcome I like, right? I can take my genome-wide data, whatever GWAS results I'm interested in, and construct this kind of predictor. I like to think of these as credit scores. So a credit score is a single number summary that's meant to kind of tell whoever's interested about your financial history, right? In the same way, we can play this game producing these kind of nice, continuously distributed things that tell us about your genetic liability for perhaps being very tall. Maybe it's heart disease, right? And so this is the kind of main tool that I use to study human genetics. Any questions? Okay, so let's now go back to educational attainment. So here's the Manhattan plot for this GWAS that was published in Nature in 2016. Remember, they have this kind of 74 loci thing, and so what that means is essentially they have 74 individual towers. They have 74 skyscrapers, as it were, right? So that's nice, but what we're going to do is think about collapsing all that information into this kind of nice predictor of educational attainment and start to ask some kind of challenging questions about it. It's not clear to me what this means. Educational attainment is a weird phenotype. It's highly contextualized by kind of when and where you grow up. There's a lot of noise in here. This is not height. That's going to be one of the kind of themes. And this is not height in ways that have kind of predictable downstream implications. So let's first just talk about prediction, right? So now we're just going to take the polygenic score constructed to predict educational attainment and think about how well it predicts that outcome, educational attainment. So they're about to publish the third version. The Nature paper was the second version. They started in about 2013 with a paper in Science. At that point, they could predict about 1% of the variation in educational attainment from measured genotype. In 2016, this went up to 4%. They're now up to 10% in the paper that's coming out. This will probably continue to grow. 10% is in the data that they have about what you can predict in terms of variation in educational attainment with also SES, so household SES, how long your mom and dad was in school, for example, and cognitive measures that they have, right? So some of these data sets have cognitive measures, and using those, they predict about the same amount of variation in educational attainment. So this is starting to get to be a pretty large number. It's basically the kind of variance that we can explain using, I would say, kind of the best measure we have from social science, socioeconomic status, or the best measure we have maybe from psychological science, something about cognition. So this is fairly large. And all prediction is done kind of out of sample, so this is well-formulated prediction, and this has now been done over numerous birth cohorts, different continents, etc. This is interesting. It's also the case that this score predicts differences between siblings. And this is a really important test. As I mentioned earlier, siblings don't differ in terms of their social background in the same ways that unrelated people do. And certainly we don't expect the genetic difference between siblings to be associated with differences they had in their home environment, right? And so it could be the case that two kids, maybe one kid born before the parents won the lottery, one kid born after. Those kids did have kind of manifestly different home environments growing up, perhaps, but that should be unrelated to genetic differences between those two siblings. And it turns out that this kind of predictive game also works between siblings, right? So you can take siblings, genotype them, and the sibling that has the higher polygenic score for educational attainment will tend to also have more education. The people that started to do this were very interested in this idea of prediction because they think it'll be useful for a variety of reasons. For example, they think that being able to have these kinds of predictors will greatly increase the power of experiments such as the Perry Preschool Project, right? They think that they would be better able to detect effects in something like the Perry Preschool Project if they had these kinds of predictors. That's it's kind of two claims. One, it's a claim about whether they can actually do it scientifically or technologically, and then there's also a claim that they can actually derive some benefit from that project. I'll leave that to them. What I want to do is start to ask some more complicated questions about this kind of prediction. So one thing you might ask is, well, what is driving this, right? Why is it that people that have higher kind of scores on this variable end up with more years of educational attainment? One thing that's been noted is that people that have higher marks on the scores, these blue bars, they basically very early in life start to kind of have noticeably superior performance with various kind of verbal fluencies than do people with lower scores, the red bars. This is from age 7 to about 18. You can kind of see the difference there. In this study, they also looked at a variety of milestones that kids had hit by age 3. Kids that had higher marks on the polygenic score for educational attainment, they weren't sitting up earlier, they weren't potty training earlier, they weren't feeding themselves earlier, they were talking earlier. That was the only thing that we really could observe in terms of the difference between high and low score people. So this is some kind of, I think, corroborative evidence that we're actually picking up something that's meaningful and not purely kind of a byproduct of something else that we're interested in. This is work that we've been looking at where we're actually interested now in whether or not having a higher mark on the polygenic score produces you to be predicts you being upwardly mobile within your own life. So each of the 4 panels is a different data set. So down here we have the health and retirement study. This is a national representative sample of Americans. This is the Wisconsin longitudinal study, also known as the Happy Days study. So if you ever watched Happy Days, that was about a group of high school classmates. There was a 1/3 sample of that high school class in Wisconsin, which is where Happy Days was filmed, that was impaneled in the Wisconsin longitudinal study. This is Dunedin. This is a birth cohort from the island of New Zealand. And finally, we have Ad Health. This is a nationally representative sample of students who were first impaneled when they were in secondary school. They've been followed up until about their mid-30s. What you can see in each of these cases is when we divide people up into those who have relatively low, middle, and high socioeconomic households when they grew up, and then think about where they ended up in their own life, that's the y-axis. More educational attainment-related alleles predicted higher ending point controlling for your starting point, right? So those who had the highest marks on these polygenic scores tended to in their own lives, accrue the most school, money, job status, etc. Right? So again, this, I think in the context of a validity study, is important evidence that we're actually getting signal here and not noise. You can certainly imagine the case that the GWAS would have just picked up things associated with kind of ancestral privileges or something like that. And then you wouldn't really get these kinds of findings, I don't think. You wouldn't necessarily see that people that were born in high SES households, they would probably stay high SES, but they would not necessarily outpace those high SES peers who just had a lower score on this genetic measure. Okay, so maybe at this point I've convinced you that prediction of phenotype from saliva is possible. Maybe not. But I think this will get interesting now. So now I'm going to start to look at the various things that are actually happening in human lives that really make this messy. So I want to talk about 2 things. I'm going to talk about the first very briefly, issues of selection, kind of something near and dear to the heart of social scientists. And then I'll go to social context. So one thing to note about all of the genetic studies that have been done is that they're really the end product of a whole number of selection processes. We've got well-documented demographic selection into these data. We've got selection as a function of health. That's what I'm showing here. So in each of these panels, the gray bar is the people that got genotyped. The black bar is the non-genotyped people, and we're conditioned on birth year. And what you can see is that the non-genotyped people are more depressed, have higher rates of heart disease, have worse health, and have more diabetes than the genotyped people. Right, so when we are studying genotypes, we're studying not random sets of genotypes, but substantially selected sets of genotypes. And this is, I think, very important. There was another study that was done showing, for example, that those who are more genetically predisposed to schizophrenia are much less likely to be enrolled in genetic studies of schizophrenia, which is not that surprising if one knows about schizophrenia, but those kinds of selection pressures are really important, poorly understood. The biologists don't think they're relevant. I think that's crazy. This is, I think, one reason or one area where more social science is kind of desperately needed in the biology. Another thing that I'm very interested in is the idea of the social genome, right? So basically, the question is, what are the role of genes not held by you, but held by people in your social sphere? I first became aware of this due to a mouse study. So it turns out that, they basically take mice and torture them, which I never fully realized, but they make them like swim for as long as they can, they punch holes in their ears, they get all these kinds of very nasty phenotypes. And what they discovered is that the phenotype held by a focal mouse depended upon the mice in the cage with them and the genes of those other mice. And so I thought this was fascinating, and I started wondering whether something similar was happening in humans. In particular, I'm interested in whether the genotype of one's social alter may be predictive of one's own phenotype. And so I'm going to now spend a few slides walking you through some evidence from a paper that was published in January. I'm going to think about schools. This is data from Ad Health, which is a sample where people in the sample first came from schools. So we have, a focal individual here, And we also have information about the people that went to school with him. And so I'm going to think about the relevance of this guy's own genes, but also the genes of the people in the school that he attends. And I'm going to do even better. AdHealth not only has data on who you went to school with, but has data on who you say you're friends with. And so we're going to think about just those people in your friendship network and think about the role that plays. So the basic question I want to ask is whether the social genome, right, this kind of aggregate measure of the genome based on the people that you're in social contact with, whether that predicts an individual's phenotype. I'm going to use a few. I'm going to use educational attainment, BMI, and height. And I'm going to ask whether the social genome predicts individual phenotype and its own genotype. So very simple. Have your own phenotype, and I'm first going to control for your own genes, and then I'm going to look at these two measures of the social genome, friends and schools, and see what we find. Okay, so here's the picture. The gray bar in each of these panels is how predictive your own genes are of the outcome. So you can see we can get pretty good prediction from your genes of your phenotype. That's the gray bar. I can predict how far you went to school based on your own genes. That's what I was talking about in the previous slides. The blue bars are the social genome measures. You can see the school is a little stronger than the friends, but in either case, we have substantial predictive power from the social genome above and beyond what we had just from the individual, right? So let me put some numbers to this. A 1 standard deviation increase in your own PGS, in your own polygenic score, predicts about an extra 0.6 years of schooling. A 1 standard deviation increase in the social genome measures, predicts an amount of schooling, an increase in schooling of, a similar magnitude, right? This is maybe about half, this is a little more. So that's quite provocative. It's also the case that we actually get some attenuation in that top bar, the top gray bar, when we include the measure of the social genome. So the red line, that's just how much your own genes predict when we don't include measures of the social genome. And what you can see is that might be an overestimate. If I'm only interested in kind of within-person processes, I'm overestimating how far, I'm overestimating things. My estimate of genetic effect is upwardly biased. And you can see the kind of shrinkage there when I introduce the measure of the social genome. Now, in contrast, how do BMI and height look, right? How heavy you are, largely independent of the genes of the people you're around. How tall you are, certainly independent of the genes of the people you're around, right? This is not surprising. I mean, we think about height as being largely a within-person phenomenon. Educational attainment is just not the same, and we're really kind of seeing that. And this is important because the GWAS technology used to study educational attainment is the same exact thing that they used to study height and BMI. They took that and just kind of transplanted it. And we're now seeing the kind of, the very kind of qualitatively different flavor of the findings that result. So just as kind of a quick summary of those slides, I didn't talk about this, but we know that there's some kind of genetic clustering in schools and friendships. We also have evidence that your social peers influence your educational attainment. And so we have this kind of clear possibility for bias. Traditional GWAS has just looked at this black to black, the person phenotype to person outcome link. But what we have here is evidence of this red path. We have kind of genetic correlations between your genes and the genes of the people in your social sphere. And then we have those genes predicting your outcomes, right? So there's this kind of clear possibility of bias. Or at least, it's just going to be the case that we're getting more than just the within-person part related to genes. Whether or not that's desirable, that's kind of a question, but this is of a qualitatively different nature than what we saw with height and BMI. Questions?
Has there been any studies that look into the expression of these SNPs when? Yeah. Basically, I'm asking about epigenetics.
Yeah. So that's a good question. So the epigenetic stuff, there certainly has. Epigenetics, I would say, is largely, it was very hyped for a while.
Right.
We're now finding that it's much more difficult than we had thought. Smoking leaves very profound effects on your epigenome. Everything else is much mushier. Smoking and other kinds of profound developmental insults, so for mom smoking, in utero, that kind of thing will have effects. People are looking at that. It's complicated. There's some interesting stuff starting to come out. I would also say, though, that a lot of that stuff is now thought to be under genetic control. So you have to actually understand the genetics before you're going to well understand the epigenetics.
That makes sense.
But if you're interested, I can send you some papers on that.
Sure.
Okay, so I looked so hard for a picture of mom jeans that didn't have a person in them, but I couldn't find one. But just imagine there was this cool picture of mom jeans here. There was also a kind of similar game played by a research team out of Iceland. This was published in Science just a few months ago. And what they showed was something very similar. The genetics that your mom transmits to you are important for predicting your own educational attainment. The genes your mom the genes your mother does not transmit, right? The part of her genome that you do not end up with, that also ends up predicting your own educational attainment, right? So this is more kind of observational. This is good evidence that the genes of your social environment have real influences for you. Okay, so let me kind of start to wrap up. I've largely talked about educational attainment, and that's because educational attainment is the kind of thing that has been on every intake form of any biomedical survey ever done, something that's easy to measure and they ask you about it routinely. But we're starting to see people play this game with all numbers of traits. The guys at the top are talking about intelligence, another GWAS from people looking at the autism spectrum, GWAS for ADHD. These are all from the last few months. This is like a kind of tsunami of these types of things. So even if you're not interested in educational attainment, this game is probably going to be played soon with the outcome that you care most about in the not so distant future, right? So I do think this is something that it behooves us all to know a little more about, especially if we study the kinds of behaviors that may be influenced by a person's biology. And here's where I'll really start to kind of slow things down. For those of you film buffs, this should be familiar. This is, of course, the famous standoff scene from The Good, the Bad, and the Ugly. And so that's how I'm going to break down my kind of conclusions. So let's start with the good. So I do think genetics offer potentially a powerful platform for studying human development. I think that there's potential for both early intervention based on genotype and also the development of better interventions. You know, one of the things that I think has become clear in a variety of ways, including like the replication crisis, is that we don't understand human behavior and development as we may have thought. And I think as we understand it better, we'll be better able to design interventions to kind of do the things we want. And I think genes might be able to play a role there. I think that genetics also allow for study of things that are hard to study, like peer effects. Peer effects are challenging to study because there's so much endogeneity. With genes, we know that your friends do not cause your genes, right? That's something that we can hang our hat on. That might be very useful. There's an Ember working paper on this idea if you are interested in that. And then, as was mentioned, epigenetic mechanisms are of kind of great interest, but they also, I think, will need to be studied with information that we derive from genetic studies. And hopefully I've kind of given you some idea about why I think education specialists and social scientists really have insights that I think are germane to the larger kind of biological and genetic debate. I think the things that we know about studying humans, especially humans in the context of schools and learning interventions, are really sorely needed. Okay, now the bad. So one possibility is that genetically informed interventions may offer benefits that accrue to those who can afford them, right? So I don't think this is unique to genetics. I think this is just kind of a general thing that expensive technologies will first be utilized and deployed by those who can afford them. Genetics certainly fall into that category. There are also, I think, uglier scenarios that one can start to imagine. My colleague Dalton Conley wrote a book where he discussed ways in which you can imagine people starting to make fertility decisions based on genetics. There are kind of companies starting to purport to do this. There are countries that are much less squeamish that are very much interested in doing this. I don't think this is good. I do think it's something we need to kind of think through carefully. And in particular, we need to, I think, be able to offer kind of context to help people better understand what we know from the genetic studies, but more importantly, what we don't know. So I think there's so much of that, and left to their own devices, the geneticists won't be able to tell that part of the story. Okay, so finally, the ugly. So first thing is that the vast majority of what's been done with people from European ancestry. This is a huge problem. NIH recognizes this, that people are really kind of trying to address this. I don't want to downplay it, but hopefully this will become less of an issue as we move forward. I think that the kind of absolute worst case scenario is the possibility that people could make arguments about genetic variation being tied to observed group differences. As I mentioned in the original slides, I think genetics are a really kind of core tool to utilize to try to understand why people that kind of start in manifestly different settings don't end up similar. Unfortunately, I can't stop people from doing that. So I think this is likely to be something that, well, this is going to be an area for I think, great kind of contention. There was a New York Times op-ed by a Harvard geneticist a week or two ago. Geneticist is David Reich. I think gestures in the ways in which this battle might be contested. I would suggest taking a look at that if you're all interested. I'm happy to talk more about some of these issues, but this definitely kind of keeps me up at night. To the extent that you're interested in some of these ideas, I've had this pre-print that's available under review. It's written by two wonderful grad students, Daphne Marchenko and Sam Trejo at Cambridge and Stanford, respectively. And this is on the Social Archive, so you're more than welcome to download it and take a look. Thanks again for inviting me, and I'm available for questions should you all have any. I'm also willing to believe I answered all your questions.
I think I'm not sure if I fully understood the talk, but my understanding is like your research is based on this fact that you like certain. So this the first paper that you showed that educational attainment is somehow in the genetics.
Yeah.
That's your. You base your work on that.
I do a lot of work that is downstream from this study. It kind of treats this study as not as true, but basically it's an attempt to kind of decide how much weight I think these findings actually have.
Okay, so because I just have this like uncomfortable feeling because so I checked this paper out and then in the figures they have one figure where they show like the genes that shows up in this study, the 74 loci. A lot of them are the same as genes that showed up in like autistic spectrum disorders or like learning disabilities or stuff like that. Yeah. And I just feel like how much does that say to educational attainment? You know, if people like genetically they have some, you know, like let's say learning disability, and then this study shows that okay, they might not be so high on educational attainment. How much does that say to like the school, the system, and all that, rather than genetics? Do you know what I mean?
Yeah. So if your kind of question is, what about the role of social forces? I absolutely believe those are relevant. Like when I first kind of started doing these follow-up studies, I thought it possible that we would kind of find no corroborating evidence that this was predicting things that we might find interesting. But you know, to the extent that they're discovering genetic variation that's also tied to autism, that to me is kind of striking and supports their their initial hypothesis. So I'm not. Was that a satisfactory answer, or do we need to?
I just feel like I don't feel very comfortable like using this as a standpoint to have a measurement for educational attainment.
Like why educational attainment? Like why did they start with that?
Like using genetic data as a starting point for like studying educational attainment, and I just personally I don't feel very comfortable using this as a starting point. So I think, I think there are a variety of stances one can take. At the very least, if you really disagree with this, like this is published in Nature. This is, I don't think you can just ignore this away. Like if you really disagree with this, then to me, like it behooves one to speak most loudly back against it. And so
I just don't know how to ignore this, I guess. It's not that I like think this is what we should all be doing or where we should all start. I don't know how to ignore it.
Okay.
I think what might be the concern here, and what I was wondering as well, is that educational attainment is sort of this societal, you could say, social construct. It's just something that we made up. It's like saying, who likes the color red? What are the genetics, right? But you had shown that talking early is the thing. So what is the correlation between these 74 chromosomes and talking early? Is that just the thing that those 74
Yeah, so
alleles-
basically, so let me just restate the kind of thing I said
Sure.
here, that when we kind of collapse the genetic predictor down based on the entire set of results from that GWAS, we see that people that are higher on that score, you know, basically talk earlier and are kind of more
I
fluent
see.
throughout their school and careers.
Okay.
That's right.
So you said it was primarily based on a European sample. And one of the things that we know about Europe is that there's often multiple language fluencies there. So I'm wondering, one, you know, how that might factor into the notion of, you know, higher fluencies, because you have, you know, all these multiple languages, and therefore, how would you translate that to a setting like the U.S. where we're so monolingual?
That's a fascinating question. Empirically, it is the case that the results from the GWAS that does have a lot of European samples predicts in the U.S. But I do think you're asking in some ways a deeper question about what are the implications of the kind of, you know, potentially unique facts about Europe that maybe were uncovered therein. And I don't have a good answer to that. I'm gonna have to think more about that. It's a very interesting observation.
So let me just extend the question into the issue that you were saying that it hasn't really been tested in context where there's maybe extreme variability, because, you know, if you're in Finland or Denmark or Sweden, Norway, Iceland, you can go, you know, to the European countries and the European Union, except in just a few, you find you know, maybe much more of a genetic consistency.
Yeah, very
In
homogeneous
terms of all the things identified ancestry, parents, you know. So, and in the United States, with immigration and with the range of diversity, is your argument that it's still translatable, you know, at a high level of reliability to the context that we, you know, experience here?
Well, so for example, in the siblings in both AdHealth and the WLS, we can see that, you know, the polygenic score, differences in the polygenic score predict differences in sibling attainments. So it does seem to be the case that we can kind of do the prediction amongst relatively homogeneous groups from the US. This group in Wisconsin was, you know, from the origin largely just a bunch of white people of Scandinavian ancestry. AdHealth is more diverse, but in this study we're restricting it to those of European ancestry because of the underlying kind of GWAS technology. We have also looked at how well you can predict in non-European samples, and you know it's the signal is substantially attenuated, but you still do get some kind of predictive power. It's just at much lower levels, as would be expected given kind of the technical difficulties that are underlying the entire enterprise.
Can I ask just one last question, just from
Of course.
because I have twin daughters who are not identical. Has any work been done in relationship to the issues of predictability with sets of twins who are not identical? So that the question of their attainment would be tied to their genetics as opposed to their social.
So I would say yes in both directions. So, you know, all of the classic twin studies were done comparing identical twins to fraternal twins. And so the basic observation there is that pretty much independently of what you're looking at, you see that identical twins tend to be more similar than fraternal twins. And to the extent that identical twins are more genetically similar, that's the underlying kind of basis for heritability estimates. People have also started to study, you know, measured genetics in fraternal twins, and a lot of the things, like for example, we have another data set that I'm not showing here that has fraternal twins, and you can kind of predict with this genetic score height differences, education differences, et cetera, between these fraternal twins.
I was wondering if you could comment on, because this is kind of a new field or a new way of asking questions, are there things that you're sort of excited about or anticipating for the next couple of years, like where you hope this might go, or questions that you're really excited to look into?
Yeah, I can imagine scenarios in which, so for example, as I understand it, we largely diagnose dyslexia too late to perform the most effective interventions. I can imagine a scenario in which we come up with some kind of genetically informed test for dyslexia. It might not be easy, and like there are kind of technological hurdles we have to get over, but that seems like a plausible kind of outcome. Outside of that, I mean, in education, I don't think, I mean, I guess if I'm being honest, I think most of the consequences are probably more wide. I think there are some potentially optimistic ones, some uses in social science, but then there are a lot of things to worry about. So, I mean, yeah, I don't know, like what you do with that, like, and I guess in some ways I kind of think about myself as like playing the role of policeman and trying to keep the geneticists honest. But yeah, I mean, you know, the Chinese, for example, are kind of very not shy about just, you know, talking about, you know, they're CRISPRing babies, right? So CRISPR is this technology to go in and edit SNPs at the you know, level of the base pair, and they're very interested in kind of CRISPRing babies to make them smarter, right? Like, what? I mean, that's something. That is that requires like some kind of serious thought and pushback, I think. And so, yeah, I don't know that there's a lot of upside to this stuff for education in some ways, but that doesn't make it less, I think, relevant or important.
I have one last question. And you talked about your idea of reducing all these variables to a single score, like a credit score. I must have missed what the implication of that move is for making this, you know, orientation to genetics more something. What is it making it more to reduce it to a single score?
Well, so it simplifies our lives substantially, that we don't have to think about adding 1 million predictors to our models or to our kind of, you know, attempts to understand the world. We just need to add this one thing. And given that we've largely uncovered the fact that most traits of interest are driven by, you know, small effect variants across the entire genome, this is kind of a theoretically coherent thing to do. And so you can already, you know, if you go and do 23andMe or something like that, they're going to provide you information about your value on a bunch of different polygenic scores. They're largely going to be kind of goofy ones, like whether or not you like sweet or sour stuff. But you're already seeing this kind of idea start to filter into you know, direct consumer genetic testing. Like I said, it's something that people really want to think about doing for heart disease, for type 2 diabetes. Like, this is something that the medical community, I think, is starting to embrace. And so that to me makes it highly salient, especially since many medical phenotypes have educational implications.
So if it's reduced to a single score, is the score ever changeable, or once it's
So
assessed then it's permanent? That's
Yes.
you for the rest of your life.
That is correct.
27 or.
That is correct. And so I mean, that, you know, in some ways is kind of, to the extent that we want to design better interventions, this might seem disheartening, but I think what it speaks to is that if we really can understand and follow, for example, what it is about a person with dyslexia that causes them to struggle with reading, we will be able to better design interventions that can kind of ameliorate those differences and are targeted on kind of shrinking those differences, you know, from the jump. We won't have to wait to observe it and then put an intervention in place. We won't be trying to come up with interventions based on our best guesses of what things are underlying the kind of development of dyslexics problems. I think we'll be better positioned to, you know, create more targeted interventions.
So if we're interested in coming up with useful interventions like that, and not interested in CRISPRing babies or, you know, a lot of fertility decisions based on this sort of stuff. Is the research going to be different for those two general kinds of goals? Are there characteristics that research could have that would favor one of those over the other?
That's a really good question. That's a really good question. I don't know that I have a great answer. I mean, you know, one thing that strikes me as a hallmark of research in this vein that might be informative, maybe it doesn't kind of lead to direct use, but it's at least informative, is research done using genes to understand, you know, why people that we don't have other good reasons for thinking should be different end up different, right? Like, you know, to the extent that we're talking about kids in a school, some of them develop reading problems, some of them don't. There's no kind of obvious stratifying factor across those kids. Genes are a kind of plausible mechanism there. So studies like that, I think, are potentially informative. You know, anything that starts to attempt to utilize genes to explain why kind of groups differ, where we kind of have an idea about what the group distinctions already are, that is much more kind of upsetting, potentially problematic, I would say. Should we call it a day?
Thank you very much.
Yeah.